go ahead and get started today. There's still folks slowly coming in, and that is perfectly okay. Well, for those of you who are here, I would like to welcome you to the Prairie Reconstruction Initiative Winter webinar series. This is the first of 3 talks presented by researchers engaged in the science and practice of prairie reconstruction. My name is Jamie Ellis, and I work as the Natural Areas Coordinator at the University of Illinois, Urbana-Champaign, and I serve on the Prairie Reconstruction Initiative Advisory Team. So I thank everyone. I thank you all for joining us today. So this session is being recorded, just for your notice, and will be available through the PRI website and YouTube, hopefully within the next month. I kindly ask that you keep your connections muted during the session. And if you have questions during the talk, I ask you to please type them into the chat, and I'll read through them at the end of the talk, and our speaker will do their best to answer them for you. So before I introduce our speaker, Becky Esser will tell you a little bit about the Prairie Reconstruction Initiative. Becky?
Thanks, Jamie. So my name is Becky Esser. I also serve on the Prairie Reconstruction Initiative Advisory Board. I'm a biologist with the U.S. Fish and Wildlife Service out of Detroit Lakes, Minnesota. Just a little bit about Prairie Reconstruction Initiative if you're not familiar with our collaboration. PRI is a community of prairie practitioners and researchers from more than 30 organizations who work together to improve prairie reconstructions so that they're biologically diverse and ecologically functional and resist invasion by non-native plants. We focus our activities within 2 general areas, evidence-based tools and peer-to-peer learning. The PRI tools include a vegetation monitoring protocol and a renewed and hopefully about to be released prairie reconstruction database for collecting standardized suite of data about our prairie reconstructions. By pooling and analyzing data from many reconstructions, we hope to reveal those pivotal elements that will increase the likelihood all our reconstructions meet expectations. Our learning opportunities include both in-person and online events, such as this one today, where the PRI community can exchange experiences and knowledge with each other. We believe these insights from both individual experiences and scientific investigations lead to our collective success. And now I will turn it back over to Jamie to introduce our speaker for today.
Becky, thank you very much for telling our audience about the Prairie Reconstruction Initiative, and either Becky or I will put a link to the website in the chat. And now I'm very happy to introduce Dr. Anna Peschel, and I'm going to stop sharing my screen so she can share her screen with us. Dr. Peschel is currently a postdoctoral researcher for the Healthy Prairies Project in the Department of Ecology, Evolution, and Behavior at the University of Minnesota, Twin Cities. Her talk today is titled Investigating the Scale of Local Adaptation in Minnesota Tallgrass Prairies. So, Dr. Peschel, thank you very much. The floor is yours.
Great. Well, thank you for the introduction, and also thank you so much for the invitation to your seminar series. I'm really excited to present some research from the Ruth Shaw Research Group at the University of Minnesota, where we are investigating local adaptation of native plants in the Minnesota tallgrass prairie. So first, I'm going to share results from the Healthy Prairies Project, and this is funded by the Environment and Natural Resources Trust Fund of Minnesota. This project is led by Drs. Ruth Shaw and Georgiana May, and the work I'm going to present is in collaboration with Drs. Shelby Flint and Naomi Rushing. So one of the aims of this project is to evaluate how distance affects population fitness for Minnesota tallgrass prairie perennial plants. So we're really interested in evaluating if plant population fitness declines as seeds are moved and grown away from the population's home site. Then I'm going to present part of my research that I did for my dissertation using the annual prairie legume Chamaecrista fasciculata, or partridge pea is its common name. And here I investigated the capacity of this plant to adapt to climate change in its home site without introduction of seed from other populations. And this is to kind of provide a counterpoint to the idea that we need to move seeds around in order to facilitate adaptation to climate change. So before I dive into results, I want to go over what I mean by local adaptation. So local adaptation occurs when the fitness of a population is greater in its home environment than when it's planted in foreign sites. So here we're defining fitness as the total number of seeds that a plant produces over its lifetime. And local adaptation has been found to be widespread, but not ubiquitous among plant species. And the scale of local adaptation is still not well known. So looking at this figure, this is from Gorton et al., which came out in Evolution in 2022. We can see different idealized examples of the scale of local adaptation. So if we consider 0 to be the location of the restoration site, local adaptation for a species occurs if fitness declines as populations are sourced farther away from the site. And this could really be steep or shallow. There also may be no local adaptation, and that fitness is the same between populations no matter how far away they're sourced from the home site. And then in contrast, a species may be maladapted to their home site if populations sourced farther away from the restoration site have a greater fitness than those that are sourced close by. So this is showing absolute distance, the figure on the left, and absolute distance is how we always tend to think about geographic distance. But in this era of climate change, it may make a big difference if seed is sourced, 200 kilometers north versus south. So the plot on the right is showing directional distance. And this could be calculated as, for example, the latitude of the source population minus the latitude of the garden where that population is planted. And this really lets us see if there's a directionality to the local adaptation pattern. Some studies have suggested that local adaptation for species may be disrupted by climate change. And an example of what that would look like is shown here in the figure on the right. So the black line shows that at a restoration site, so that 0 kilometer mark, those populations sourced from warmer sites farther away from the south, they may actually have a higher fitness than those sourced closer to the site, or those sourced the same distance away from the south as to the north. I will point out that this is an assumption when we think about climate change adaptation, in that we should source seeds from the south or more southerly, warmer climates, but that they may not always have greater fitness, and this really bears investigating. Many of you are likely familiar with this map. It's from the Minnesota Prairie Conservation Plan, and it lays out the goals for converting and restoring tallgrass prairie in Minnesota. And one of the goals is to restore some 300,000 acres in the native prairie core areas and creating corridors of tallgrass prairie between them. So there are seed sourcing guidelines for prairie managers, but they do vary by agency. And we still, even though we have some guidelines, we still don't have much information on the scale of local adaptation for perennial plant tallgrass prairie. And we really need these in order to ensure that when we go to do restorations or management, we're fostering robust and persisting populations. So the Healthy Prairie Project is really seeking to investigate some of these outstanding questions of local adaptation in the tallgrass prairie ecosystem in Minnesota. So for this work, we evaluated local adaptation for 4 prairie perennial plants. They're shown here, listed by their common name, and then underneath their 4-letter Latin code name. And I'll be referring to both of those throughout the presentation. We also have data on 2 grass species, side oats grama and little bluestem. And those data are currently being analyzed by our colleagues, Shelby Flint and Wes Breaker. This is also likely a familiar map to many in that it shows the extent of the tallgrass prairie in Minnesota. So the historic extent of the tallgrass prairie is in yellow, and then the remaining remnant populations are in red. So to evaluate local adaptation for these 4 species, we collected seed from 12 populations for each species across this prairie region. So in the Northwest, the West Central, the Southwest, and then the Southeast. And we had a minimum of 3 populations from each region. And then within these regions, we collected seed from scientific and natural areas, and those have native remnant prairies in them. With these seeds in hand, we propagated them in the greenhouse. And then when they were mature, we planted them in a common garden at the Rosemount Research and Outreach Center, which is shown here in purple on the map. And that's about 40 miles south of the Twin Cities. So the purpose of the common garden at Rosemount was to attenuate maternal effects. So these are aspects of the maternal plant's environment, so the seed-producing plant's environment, that affect the phenotype of their offspring or of the seeds. Our original seed collections were from natural populations, so they could have experienced different environmental conditions in the year that we collected seed. So for example, perhaps one population received copious amounts of rain that could cause the maternal plants to produce very well-provisioned seeds, which cause those seeds to be very vigorous compared to potentially seeds from another population that received very little rain. So by planting all of the populations together in a common environment, this can lessen the effect of environmental vagaries and really allow us to better evaluate plant population fitness based on their adaptive evolution to their home site. So at Rosemount, we had 12 plots. An example of what one plot looks like is shown here on the left. And in each plot, there was one population for each species. And we did this to prevent mating between populations of a species from different sites, from different populations. And then we collected seed from these plants at Rosemount, which were then planted into our common gardens in Western Minnesota, where we evaluated local adaptation. So in the map on the right, you can see the location of our common gardens in orange. We really tried our best to find sites that were similar, but they did have differences, particularly with gopher activity and the composition of agricultural weeds. However, all of these sites were prairie restorations. So they had previously been in agriculture and then were being converted by the Department of Natural Resources to tallgrass prairie. You'll see a big X at the middle site. We had to let that site go, because it was getting overrun by grasses due to some particular management restrictions there. So we evaluated plant fitness and local adaptation at the remaining 2 sites. Twin Valley was our Northwest Common Garden, and then Lake Bella was our Southwest Common Garden. And we did that for 6 years. So in each common garden, there was seed from all 12 populations of each species. We tracked seeds planted and then recorded their survival and the number of flowering heads that they produced each year. So for these results, fitness of a population is the average number of total flowering heads from 2017 to 2022. So first, we evaluated if mean fitness differed among populations for a given species at each common garden. Then, if we did detect differences in population fitness, we evaluated how geographic distance between the source population and the common garden affected fitness. So again, this figure shows what we expect to see with local adaptation. in that as distance between the source population and the common garden increases, we see some decline in fitness. But we decided to evaluate geographic distance in 3 ways. So first, we calculated the absolute distance. So that is the shortest distance between the common garden and the population. It's how we normally think about distance, in that there's no negative value, just like you would see in a map. It's just the distance between 2 points. So if the population, in gray, if it was the same distance north of the black dot, the representative common garden, you would get the same distance number for those 2 populations. However, evaluating the difference in latitude can capture the difference in distance, considering if a population is north or south of the common garden. So in this graphic here, latitude is represented by the dotted lines, and the difference is by the black solid line. Here, the difference in latitude would be negative, as southern latitudes are lower than northern latitudes. But if the population, so that gray dot, was north of the common garden, the difference in latitude would be positive. And then lastly, we calculated longitudinal distance, which considers if the population is east or west of the common garden. And in this example, longitude is represented by the dotted lines and the difference in longitude by the solid black line. So this difference would be positive as longitude increases eastward. Okay, here are some results. Don't get overwhelmed. You'll see a series of figures that look like this. So I'm going to walk you through what to look out for. So this, first of all, is showing geographic distance at the Southwest Common Garden, and points are mean fitness for each of the populations. And the line is the fitness function. There are 4 rows, so each row represents a species. So thimbleweed on the top, white prairie clover, purple prairie clover, and then bee balm or Monarda, Mophie. You will also see that there are 3 columns. So the first one represents absolute distance, then latitudinal distance, and then longitudinal distance. The vertical dashed line is at 0 in each of these plots, and that represents the location of the common garden. And then for some of the plots, there is a horizontal solid line at 1, and that indicates that mean fitness of 1. So the y-axis in all of these plots is mean fitness. So a mean fitness of 1 indicates that populations are at replacement, but points above 1 show that populations are increasing, and points below 1 show the populations are declining. For some of these species, their population mean fitness was so far below 1 that it's just out of the range of the y-axis, so you don't see a solid line at 1. And then the points are coded by region based on color, which you can see in the legend on the far right. And then for latitude and longitudinal distance at the top, I've put some keys so that you can see if populations are south or north corresponding to their latitude, or if the populations are west or east of the common garden. So first of all, for Dapu, or purple prairie clover, we did not detect that distance was a significant predictor of population fitness. You can see that the lines are pretty flat and there's a lot of scatter. For the other 3 species, we did see similar trends in that populations sourced farther away from the common garden had a lower fitness. And also we found that northern populations had a lower fitness compared to those from the south, and that populations sourced east of the common garden tended to have a higher fitness than those sourced west of the common garden. So now a similar set of figures, except these are our findings from the Northwest Common Garden. And we found different trends at the Northwest Common Garden than we did at the Southwest. And we also found that the effect of distance on fitness tended to vary between species. So first of all, ENC or thimbleweed is not shown here because fitness was 0 for this species for all populations in the north. And also in this common garden, we found that these distance metrics were not a significant predictor of fitness for DACA, which is white prairie clover at the top row. We did find differences between Dapu and Mofi, in that for Dapu, fitness tended to increase as populations were sourced from the south until a point, and then fitness tended to decline. And then we also found that fitness decreased as longitudinal distance increased. So eastern populations tended to have a lower fitness for this species. Whereas for Mofi, fitness continued to increase as populations were sourced from more southerly populations, and fitness tended to increase with longitudinal distance. So geographic distance is a pretty easy and straightforward measure to use when evaluating local adaptation, but it doesn't always explain it. So we also decided to evaluate differences in climate variables between the source populations and the common gardens. We calculated differences in mean monthly temperature and total precipitation over the growing season between the source population and the common garden. And so for the source population, we calculated these historic averages for these variables, so over a 30-year period. But for the common garden, we calculated these climate distance averages over the course of the experiment, so when the seeds were planted in the site. So you'll see a very similar series of graphs to the distance graphs, except this time precipitation and temperature are on the x-axis. And again, on the top of these graphs, I've put some keys as to if the populations are warmer, colder, or drier or wetter compared to the common garden, which is again at 0. So this is showing climate distance at the southwest common garden. And when evaluating climate distance at the southwest common garden, like we did for distance, we saw similar trends among species. Primarily that fitness increased as precipitation and temperature distance decreased. So source populations from wetter and warmer sites, those which were more similar to the climate of the southwest garden, had a greater fitness than those that were sourced from drier and colder sites, which were more sort of dissimilar climatically to the southwest common garden. And so now here, looking at the relationship between population fitness and climate distance at the northwest common garden, similar to when we compared distance differences between the two, we find different responses at the northwest than at the southwest common garden. Again, as with distance, climate was not a significant predictor of DACA fitness here. And then with Mofi and Dapu, we found that source populations from wetter and warmer temperatures, so those that had a greater climate difference to the common garden, had a greater fitness. However, the bell-shaped curves with Dapu means that fitness increases as climate distance increases to a point, but then starts to drop off. So there seems that there might be a shift in this local adaptation curve, which is not a result that we saw in the southwest common garden. So what can we conclude from the geographic and climate distance predictors at these two gardens? I think our data suggests that northern populations are showing signs of maladaptation at the northwest common garden. And this is because fitness at the northwest common garden was greatest in populations that were farther away geographically and climatically. Maybe this suggests a disruption in local adaptation from climate change. But we don't really know if these climate variables are causal. We could be seeing these trends from some other factor that we didn't measure that might be correlated with these climate variables. But I think the data suggests that there is some local adaptation for southern populations at the southwest garden, because in the southwest garden, populations that were more similar in geographic and climate distance had a greater fitness than those from more northern latitudes, so farther away geographically and climatically. So, however, while we did detect that there were significant effects of climate and geographic distance on population mean fitness, they are not universal in predicting population fitness. So what I mean by that is this graph here shows the fitness function for thimbleweed at the southwest common garden for latitude distance. And we can see clearly that in general, as fitness declines, fitness declines as populations are sourced from more northern latitudes. However, there is a northern population, so this northern population here in red, that has fitness that is as equal to some of these southern populations. And there is a southern population down here in blue whose mean fitness is equal to some of these northern populations. So there are trends, but there is quite a bit of scatter. And I think this shows that when sourcing seed for restorations, doing so from a variety of populations is important. Also, it's important to point out that regardless of any patterns between fitness and distance, what ultimately matters for population persistence is if population fitness is at or above 1. But we're discounting rapid adaptive evolution, which I'll talk about soon. So to clarify what I mean by this, so again, in the figures where you see a horizontal line at 1, that's indicating that populations above this line are increasing and below are decreasing. If populations stay below 1, they'll just dwindle to extinction. So here for geographic distance at the Southwest Garden, we see that there are only 2 species that had populations above 1. So that's Daca and Mofi. For Daca, there were 5 populations above 1, none of which were from the Northwest, but there were some from the West Central, and some Southwest populations are also below replacement. On the other hand, for Mofi, all but 1 of the populations are at or above replacement. So interestingly here, while the recommendation at the Southwest is to source seeds from southern latitudes, really for Mofi, these populations are growing and persisting regardless of where they're sourced from. These trends, could change. We do not have complete life history data for these species. Some of the plants are still alive and producing seeds, but this is now we're in the 7th year of this study. And these plants are not very long-lived perennials. They have a lifespan somewhere between 3 to 10 years. So while we don't have total life history data, we have a good idea of plant population trajectory in these common gardens. So now I'm going to talk about Dr. Rushing's work in the Healthy Prairies Project, where she evaluated fitness for thimbleweed and white prairie clover at the Rosemont Common Garden, looking at climate and geographic similarity. And this map just shows the distribution of the source populations that she used in comparison to the location of the Rosemont Common Garden, which is the black circle on the map. And kind of cutting to the chase, the main takeaway from this work is that for both species, fitness was greater in populations sourced from latitudes north of the Rosemont Common Garden when evaluating geographic distance. But the effect of climate similarity on population fitness was different between these 2 species. So for thimbleweed, source populations with a similar climate to the common garden had a greater fitness. And this, interestingly, is similar to what we found for our Southwest Common Garden, but not in our Northwest Common Garden. But for white prairie clover, climate similarity was not a significant predictor of fitness. And this is what we found at both of our Western Common Gardens. So these data might suggest that climate, at least for the variables measured, is not an important factor in local adaptation for white prairie clover as it may be to other species. So taken together, we see that latitude of the source population can be an important indicator of population fitness when conducting restorations, but the direction of this effect seems to depend on the restoration site. So for the Northwest and the Southwest common gardens, populations from more southern latitudes had a higher fitness. But at the Southeast Common Garden in Rosemont, at least for the 2 species evaluated, populations sourced from northern latitudes had a greater fitness. So it's a little bit of a different result. When thinking about facilitating adaptation to climate change for plant populations, we tend to assume that we should add seed to our sites from populations in the south, as those are warmer and may confer adaptation to the warmer climates predicted for our sites in the future. However, I think this shows that while we assume moving seeds from south to north will increase fitness, particularly in this era of climate change, that really may not always be the case. And I think too, this shows that populations may not always be adapted to certain climate variables that we assume they are, such as temperature and precipitation. There are many other aspects of climate and the environment that populations could be adapted to. And it's likely very challenging to figure out exactly what constitutes the environment, as it's vast and hard to quantify with a few variables. Okay, so switching gears, I'm going to show some results from my dissertation research that I did while I was in the conservation biology program at the University of Minnesota Twin Cities. And here I evaluated the capacity for adaptation of Chamaecrista fasciculata, partridge pea, in response to climate change in its home site without addition of foreign seed. So to do this, I used a space for time substitution field design. I had 3 prairie reconstruction field sites along an aridity gradient, shown here, ML, LB, and SD. So these sites get warmer and drier east to west. And I planted a pedigreed population of Chamaecrista fasciculata seed sourced from the ML site into each of these sites over 2 years. And estimates of the rate of adaptation of the McCarthy Lake population planted into these western sites will give insight into its capacity to adapt to climate change, as the climate conditions from these western sites are predicted to impinge upon McCarthy Lake in the future. Importantly, I retained a portion of the seed planted in the first year, which is called the parental generation. And I planted this generation in the second year alongside the offspring, so the progeny generation, that were produced in the field from the parental generation planted in the first year. And I did this because the difference in mean fitness between the parental and progeny generation when planted together in the second year will give insight into the rate of adaptation for this population. So that's the rate at which the population can increase its mean fitness due to adaptive evolution. Planting the generations in the same environment in the same year means that any difference in fitness between the generations can be attributable to selection in the first year. So this captures a genetic change in mean fitness, not a change in mean fitness due to the environment. So you can kind of think about it as a heritable change in mean fitness. So I'm going to show you a series of results using this figure. There are 3 rows, one for each site, ordered east to west. Mean lifetime fitness is on the y-axis, and notice the difference in scale between sites. The dashed line is at a mean fitness of 1, which again means the population is at replacement. So fitness below this line indicates population decline, and above is population growth. And this panel shows that mean fitness of the parental generation at each site in the first year. You can see mean fitness of this parental generation in each site in the first year. And it's showing that it decreased away from the home site, McCarthy Lake. So it remained above replacement at South Dakota, the westernmost site, but interestingly, it fell below replacement at the middle site. So this shows that fitness does decrease with distance, but it's not linear, as the middle site, LB, had a greater decline in fitness than the farthest site. And importantly, fitness, while it declined, is still above 1 at this westernmost site, which means that the population here is still growing. So now I'll show you results of the genetic change in mean fitness. So this is again the difference in mean fitness between the parental and progeny generation in the second year, and it gives an estimate of the rate of adaptation. So this was negative at the middle site. so the progeny generation, had a lower mean fitness than the parental generation in the second year, but it was positive at SD, the westernmost sites. So this indicates that there was a decline in mean fitness in a genetic sense in the middle site. But for SD, selection on the parental generation in the first year resulted in adaptation of the progeny generation to the environment that they experienced. And this suggests that this population of Chamaecrista fasciculata from McCarthy Lake may possess the capacity to adapt to climates predicted for the future without introduction of seed from other sites whose current climate may be similar to that expected for the future. So when thinking about managing prairies, particularly our seed sourcing strategies for mitigating population decline from climate change, this work shows that populations may already possess the capacity for ongoing adaptation to novel environments that they might experience in the future. And our work with the Healthy Prairie Project suggests that local adaptation of populations to climate and distance varies between species and the restoration site itself. So, and importantly, while we found trends, there was a lot of scatter. So there are likely other factors influencing local adaptation that we did not measure and are still to be determined. So thank you again for inviting the Shaw Research Group to present our work, and thank you to my funding sources, and I am happy to take questions.
Great, thank you very much, Anna. A lot to think about, a lot to chew on there. We do have one question that was put in the chat from Christine. How do you think soil and fungal relationships may be impacting the results of this study?
Yeah, I mean, it's likely, it's a factor of the environment. I can't, I haven't evaluated in these sites any of those qualities, but we do, our collaborator, Georgiana May, that's kind of her area, and they have work coming out which is looking at home versus away microbes and how that could affect plant fitness. So is the microbial community in the soil and in the plants that they experience in their home population, are those different than at these common garden sites? And if so, are they having an effect on fitness? So yeah, very pertinent question. And there's some research in that regards coming out.
Great, thank you very much. Are there other questions? If so, you go ahead and unmute yourself, and you're welcome to ask a question.
It's a lot of results to take in at once, so you're still absorbing the information. That's fine too.
I guess I'll speak up again. I have another question. This is Christine speaking again. One of the things that kind of came to mind while you were giving your presentation and your suggestion that having seeds and plants sourced from multiple different locations might be one way to address kind of the scatter results of your study. And one thing that comes to mind as a practitioner is that it's oftentimes really challenging to know exactly where seeds are sourced from. And that's, even true with plants. So trees, for example, we're oftentimes ordering from nurseries that have sourced those plants from who knows where. Sometimes we know, sometimes we don't. And I'm just curious if you have any recommendations for the industry and for practitioners to set some standards on how we can maybe share that information or keep track of that information a little better.
Yeah, well, I know that there is like the Yellow Tag plant program in Minnesota, which I think they do try to get more detailed information on where the seeds are sourced from. And interestingly, one other branch of the Healthy Prairies Project is just what you talked about, is providing producers with more site source seed where the source is known. So part of what we have also done is gone around and collected seed that is from species that are of interest to plant producers and growers, and we have sent out inquiries to them to see if they would like some of our seed, and we have developed some material transfer agreements and have gotten site source seed into the hands of seed producers in Minnesota. So yeah, and there's a paper that our group published in Restoration Ecology just all about that. I think it's Goldsmith et al. 2021, just about the challenges that the industry faces with obtaining site source seed and producing it. And I think that paper has some good recommendations. Yeah, I can share that. I can share that paper if I just dig it up here. Yeah.
Great, Anna. Thank you. Chris asks, how were you able to factor out the effects of a bad growing season or seasons?
Yeah, we didn't do any factoring out. I mean, we're looking at population persistence over time. So, that's just sort of par for the course, I think. So we were evaluating every year. So the seeds were planted in 2016, and every year we would go and take data on them. So that's the thing with perennials, is you can kind of see over long periods of time how they do. With annuals, it's harder because they're just at the whims of the environment in one year. But yeah, we just, we're taking average lifetime fitness for these populations over this 6-year time. So yeah.
Yeah, does that help, Chris? And also, right, bad can be subjective depending on our views as land managers or practitioners, or even from any particular plant species. So thank you, Anna. Lisa writes, really interesting, and definitely want to share with the Minnesota DNR staff, working to incorporate climate change into the Minnesota Wildlife Action Plan revision, like with Allison Carvo and others.
Mm-hmm. Great. Yeah. Thanks. Yeah.
Okay. Other questions that you want to voice out loud or type in the chat? Please go ahead.
And I will just pull up that reference and drop it in the chat. If people are interested in that and want to stick around while I find that.
Oh, that would be great, Anna. So maybe I'll chime in here and ask what avenues of research, what interests you next to continue to explore, with plants reactions responding to climate change, whether they do or not, and how we might utilize that information as, to best inform our reconstruction practices.
Yeah, I think, still pursuing this question of, local adaptation within perennial plants. We're definitely kind of not done there. So, it would be great if we could continue the Healthy Prairies project for a few more years. But I think, there's still some more data to be analyzed. One thing that came away from my dissertation that was really interesting was sort of, since I was using an annual plant, it was really the effect of environmental change between years can really change whether or not the population is adapted to the environmental conditions. It can really kind of change the dynamics of this population. So getting a better handle on for annuals, I think how year-to-year environmental variation can really influence population persistence. But yeah, I think just in general, figuring out ways in which we can manage prairies better so that we have robust and persisting populations in the future. They're so important, as everybody knows, to our migratory birds and all of the kind of water quality and soil retention aspects that they hold. So, and as I mentioned before, we're now going to be with the Healthy Prairies Project. We're going to be doing some work investigating the kind of fungal microbial aspect to local adaptation within these species. So, yeah.
Oh, very good. We definitely look forward to those results. Brandon, it looks like you have your hand up. You'd like to go ahead and ask a question?
Yeah, thanks. Nice talk, Anna. I had a quick question. You had the 2 prairie clovers, Monarda, and I forget what your 4th species was.
We have thimbleweed, anemone cylindrica, and then I should say there's work coming out on side oats grama and little blue stem.
Oh, okay. Well, that answers my question. I was curious if you're going to look at grasses and other things as well.
Those are heavy hitters for prairie restoration. So yeah,
great.
That data is collected. It's just in the hands of 2 other colleagues now. So
great. Thanks.
And then also there's another comment there in the chat as someone who has been monitoring pollinators. I wonder about any asynchrony between plant response to climate change versus pollinator response, and how that may affect floral resources going forward. Well, and then I'll also add how that might impact plant fitness as well.
Yeah. I know less about the pollinator aspect of things, but at least in terms of plant fitness, I guess we do see that, at least in the Southwest, when we did see that populations in both of these common gardens, there were populations for some of the species that were increasing and that were persisting. So, pollination is working, at least for them. I don't have concrete information. I'm sure there's some work out there that's looked at the timing of flowering and the timing of pollinator emergence and how that can maybe affect plant fitness. But yeah, that's something I know less about. But good question to raise.
Thank you, Anna. Thank you for the question, Lisa. Any other questions while Dr. Peschel is on the line? And if folks have questions later, may they contact you or the Shaw Lab?
Definitely, I'll put my email in the chat.
Okay, thank you.
Feel free to contact me if any questions arise.
Great. Thank you very, very much. Well, it looks like we've exhausted at least some of the questions here. I'm sure there's lots of people will think of other things, after we jump off the line. I would really like to thank you, Anna, for being here and presenting your research results and getting us to think even more about the complexities out there when it comes to planning our reconstructions, and especially in the in the face of a changing climate. Every bit of information helps, and I want to thank everybody that's on the line and that joined us today. Thank you. We will be back here online at this same time next week, where Dr. Elizabeth Bach from the Nature Conservancy will be talking about her work and looking at the long-term reconstruction at the Nachusa grasslands in northern Illinois. And so if there's no other questions, I think we'll call it closed today. So thanks again, everybody.
Yeah, thank you everyone for listening and for your questions. I appreciate it.